excited to see you all guys here and welcome actually to our sixth webinar already in NEMIS Academy. I can believe it because since we started this project on January, it was really promising and actually it is promising with the all exciting and great topics we gathered so far in the food safety field. First of all, let me introduce myself briefly. My name is Alina, and I'm marketing manager at NEMIS Technologies, and I will be a moderator of our today's webinar. And thank you all for joining our webinar, especially a big thanks to all who are joining the webinars from the very beginning on. And warm welcome to newcomers, and I hope you guys will enjoy today's session because today we have amazing speaker. And actually, I'm a bit nervous because we are privileged to have on board Dr. Patrick Mester, a group leader at the Christian Laboratory of Monitoring of Microbial Contaminants in Vienna. And this incredibly insightful session promises us to deepen our understanding of the critical issues in food safety and hygiene. And guys, don't be ashamed and scared so far to drop a lot of questions because I'm pretty sure you will enjoy the presentation today. And Patrick, I give you word and give your time. So you're welcome to share information about yourself and start our presentation.
Yes. Thanks, Alina, for the nice introduction, the warm words and the warm welcome. And thanks for inviting me for the speech and to give the webinar here about the viable but non-culturable state of Listeria monocytogenes in the context of food security, which is one of my pet projects I've been working on a long time. So a few words about myself. You already said I'm here at the Veterinary University of Vienna in the Institute of Food Hygiene, Food Technology. My background is I'm a classic microbiologist, but I've been working together with the food industry here in Austria and also the diagnostic companies worldwide for the last, I would say, 14 to 15 years now. We have a lot of experience with pathogen detection and regarding all the problems that bacteria, especially in the field of food production, can present not only to the authorities, of course, but especially for the food manufacturers, either primary manufacturers or even the secondary manufacturers. And as we here originally were called the Institute of Milk Hygiene in the past, you can imagine that we have a strong background, especially in Listeria monocytogenes, which is basically our pathogen here at our institute where we have the most experience. Also nowadays, we have a broader research field working with all different kinds of pathogens. So my topic today will be about the viable but non-culturable state. So I will try to share my screen from now on. I hope you can all see the presentation. So as said before, the topic today is the ability of Listeria monocytogenes to enter into a viable but non-culturable state, the VBNC state, and what that means in respect to food security and how we deal with this challenge. And as I know that the topic of dormancy, of bacterial non-culturability is still kind of a new topic and we have to wrap our head around, I will start with a brief introduction about where this topic is coming from, since when we realize it and where the current state of research here about this topic really is. Always with a focus on Listeria today, although this can be also true for many other pathogens. So the whole topic of dormancy comes when we start realizing that microbiological methods, especially plating methods, always show us the world in a black and white picture. So we either see bacterial cells as either dead or not growing if we don't find them on our enrichment media or culture plates, or as viable if they grow. And this dogma basically of either something is dead or alive based on our method of detecting them on an enrichment media has been the status quo for many decades, of course, in all of microbiology. However, we now realize in the last decades that also bacteria have a much more complicated lifestyle, and that also bacteria can stop growing without being dead immediately. And this is called dormancy. We know dormancy normally a little bit more, or we're more familiar with this topic in the context of, for example, Bacillus cereus or other spore-forming bacteria, where we are very familiar with dormancy, but this is also true for bacteria. And here I have to say that the microbiological community has a small problem, because somehow we separate this topic into two different fields, both equally important, but however still separate. And this is on the one hand called the antibiotic persistence on the left, which is a big topic of bacterial dormancy in the field of antibiotic resistance development, infection relapse, or antibiotic treatment failure in especially human patients, but also in veterinary cases. And on the other hand, the viable but non-culturable state, which is also a dormancy state, where the bacteria stop proliferating for a given time and being thus undetectable by routine media while still being alive. The only real difference between those big topics is that antibiotic resistance normally regains their culturability after a short time, while the VBNC bacteria usually need an external signal. This can be like a quorum sensing signal, a simple change in the environmental conditions. Sometimes it has to be a little bit more complicated, like an interaction with an amoeba. However, normally they need an external signal to be culturable again. So when we look back and we look at our definitions on what we are considering life or living and dead, we have to now realize that the method of choice we chose for the last many decades, especially the culture, is only showing us the actually growable cell or cultural cells at a given moment, or the dead cells. While in reality, the complex bacterial survival is much more differentiated, and we can separate into stressed and injured cells, and we have to consider persistent cells, so non-growing subset in a population, and especially the viable but non-culturable cells. And if you look at the literature, I just put a picture here on the right from a publication from 2018, we realize that all the states, however we define them, like stress, persistent, or VBNC, are probably all part of the same dormancy continuum, they call it in this article, but in the end, it's a way of bacteria to cope with stress, to survive under very challenging conditions, and afterwards to resuscitate and grow again. So we have to realize also bacteria are not just black and white creatures, which are either growing or not growing, but they can stop even the most basic viability parameter, in this case, growing. So it's much more complicated than we think. And since when do we know this, really? And here now I only focus on the topic of VBNC, because the persistent topic is a little bit older already. So this has been reported the first time actually for a pathogenic bacteria in shellfish in the US called Vibrio vulnificus, and Vibrio cholera especially as well. And what the researchers always were observing on the right was that during the cold months of the year, the cells were they were not able to detect the cells using cultural methods anymore. However, after the spring, or in the spring and the summer months, all the cells were returning, and the conclusion at the time was that the cells were actually released into the ocean and then afterwards recultivated the shellfish. However, in 1982, in the first study, and afterwards in some follow-up studies by the group of John Oliver, could demonstrate for the first time that the cells actually were never going away. They just stopped growing. And we can now create this experiment quite nicely also in our laboratories, here on the right shown. So if we store a culture of those bacteria at 4 degrees Celsius in a normal buffer system without any nutrients, what we observe is a slow but steady decrease of culturable bacteria. So here we see the classic plate counts. And as you can see, after 7 days, normally we cannot detect any culturable cells anymore, and we would have been interpreting these results for a long time as all the bacteria have died. But combining with different methods, in this case, they used microscopic methods, they could show that the bacteria were actually never gone and they always stayed intact. And if you take this culture and just store it for 24 hours at room temperature without the addition of any additional nutrients or any additional energy, the cells or the population can completely recover. And after 24 hours of incubation, we find the same amount of bacteria, meaning that the bacteria were never dead, they didn't regrow, but they simply changed their phenotype from one being able to be cultivated on a plate to one which was viable but not culturable, back to the one which is again proliferating and being culturable on a plate. So we realized that only plating and only microbiological methods really give us it's not a wrong picture, but at least not the full picture of what is going on with bacteria at any given moment, especially under challenging conditions. And since then, there has been a huge discussion about what is it then, how can we define and distinguish what is dead, what is alive. So the VBNC state here describes a status of the cell in which they fail to be grown under normally or routine bacteriological conditions under which they would normally grow. So it's an exception for the plate count. And what we consider viable nowadays is still under it's a big discussion in the scientific community. However, the minimum consensus so far is that to be considered alive, a cells at least has to be intact, capable of reproduction and metabolically active. Meaning, of course, the membranes of the bacteria cannot be damaged, at least not damaged to a degree that, for example, the whole loss of cellular energy is going on, or that genetic material is lost so that the cells couldn't be reproductive anymore. So in the end, the cells have to be intact. There have to be the complete genetic code, because in the end, replication has to be possible, which also means that there has to be at least a rudimentary or the ability to be metabolically active. So if there's no cellular energy, for example, also those cells will probably never be called alive nowadays. So here in this slide, I just want to show you what has happened since the first description of the VBNC state. So it started all with Vibrio cholera and Vibrio vulnificus especially, but nowadays this state has been described in various publications and studies for more than 100 different bacterial species, including a lot of pathogenic bacteria, but also for other eukarya, especially fungi, which makes us realize that this is not really a very unique way of bacteria to survive, but also together with the antibiotic persister topic, we realize that being dormant, meaning stopping reproduction, is just a very general stress response of bacteria to survive in either the environments or under antibiotic treatment or other treatments, and to be able to afterwards repopularize again. So this brings me now to Listeria monocytogenes, and now I want to present a little bit the information what we know about the Listeria and in the VBNC state. So as I mentioned before, the entry into the VBNC state is a stress response, and I already showed you, similar to what we've seen for Vibrio, we can do the same experiments and show that if we store a culture of Listeria in, for example, just a sodium chloride solution or at lower pH, after a certain time, either weeks or a couple of hours, we will get diverging results if we combine methods which for example, detect other viability parameters and culturability. So we see that after a while, culturability is not a really reliable indicator of the presence of viable cells anymore. And this is, of course, a problem. However, as you can see in the first slide, instead of a couple of days, unfortunately, those experiments, especially with Listeria, can take up to 10, 50 weeks. So it's really difficult to show this experimentally or to really work with those bacterial cells. However, researchers have shown this for Listeria as a response to, for example, unfavorable water activity values, pH, starvation conditions, or UV conditions. The second induction scheme is more like a really direct stress response, which is even more problematic. And we know it also from the antibiotic persisters, which is normally conditions which are not unfavorable, but are supposed to be lethal or potentially lethal stress. So here I have to mention that it has been shown in several studies that in response to, for example, biocides or biocide concentrations where we would normally believe that we would kill and inactivate all cells, those cells are actually not completely inactivated. But when we again combine methods measuring culturability on routine media and other viability parameters, we see that the actually viability reduction is much less. For example, here we show, and below you can see pictures from a publication of my group, where we studied the induction of the VBNC state of Listeria with detergents and biocides. And we could show, for example, that those cells in the VBNC state after 24 hours are almost not distinguishable from the from viable, from actively growing cultures, either by, in this case, microscopy, viability microscopy, or even using the RP strip test. So we could show also that the metabolism is still active, that fermentation was going on, and that the cells were still actively producing cellular energy, for example. And the problem here is, of course, the misinterpretation of results, because if we only rely on culturable data, often in those studies we would believe that we actually inactivated, or we found concentrations which inactivated the bacteria, but instead we only inactivated a small portion of it, while the rest remains viable and in this VBNC state. And this brings me, of course, to the relevance to the food sector, because if you remember all the conditions I've shown you before, we realize that in the food production environments, all of those conditions constantly exist. We have rapid changes between nutrient-rich or nutrient depletion conditions. We have rapid shifts in pH and water activity, temperature shifts, and also we have a constant use of disinfectants and cleaning agents, all which we know from laboratory studies can induce the VBNC state in Listeria. And which makes matters more complicated, just the fact that most of the methods we are currently using for detecting and monitoring Listeria in food production environments are at one point or the other reliant on actual cell growth and cell multiplication, especially within a certain amount of time. So without the classic overnight incubation in selective enrichment media, no method would really be working at the moment, which also means that they would not be able to detect cells in the VBNC state. So we see that potentially we can have a problem with those cells in the food production environment, but what is known about the challenges posed by VBNCs can we just consider them a novelty, like a certain phenotype which is very interesting to study but not very relevant in the food production environment or for food safety? Or how do we have to interpret this, the challenges posed by those cells? And here there has been a lot of research, not only for Listeria, and we can narrow it down to 4 different topics which can occur. On the one hand, cells, as I presented before, they retain the metabolic activity, and it is known for other bacteria than for Listeria that those cells in the VBNC state if they are in high enough numbers, can still lead to fermentation problems, especially in the juice, wine, and beer industry, but also it has been known for canned sauces or for other products that we believe that cells have been inactivated, but in reality, they can still be active enough to lead to fermentation problems. For certain pathogens, it could also present a problem if they are still producing toxins. However, this has been reported only very few times, and it's not a big problem, of course, for Listeria. The next point would be virulence. So how dangerous are actually the cells in the VBNC state? Because we still have to realize that those cells might be alive, but maybe they are so stressed that they're not really a big problem or represent a big danger for humans. And here also the research has been unfortunately a little bit scarce, especially for Listeria. But what could be shown in the past that yes, those cells still are able to infect host cells. That has been shown on the one hand in vitro models on a cell culture level, but also in a quite or relatively recent publication, for example, for this C. elegans model. So here the researchers have infected this model organism with bacteria, either in culture bacteria, which we see also on in the graph with a red line, with bacteria in the VBNC state with a green line, and with heat inactivated and really heat killed cells in the black line. And what we see that although the mortality did not increase as much as with cultural bacteria, still there was a significant difference regarding the mortality of this model when infected with VBNC cells compared to really inactivated and dead cells. So we cannot say that there are absolutely no danger. However, much more problematic is what the cells are actually supposed to do, survive. And as this is a stress response, and a very general stress response, bacteria or bacterial cells in the state can be extremely resistant, we know, to antibiotics, but also against biocides. So here's a presentation from one of our studies where we developed a method to actually be able to measure this accurately. And we could, for example, show that for certain disinfectants or biocides, such as benzalkonium chloride, bronopol, but also other biocides, the cells in the VBNC state could survive a 100-fold higher concentration compared to the cells in the culturable state. So this, of course, is a really significant and also from a practical perspective, a significant and important increase in tolerance, which is normally not known for other resistance mechanisms such as efflux pumps or other genetic modifications or factors. And this is especially problematic given the last problem connected to VBNC cells, and that's the fact that they can leave this state again. So in the end, after they survived, for example, a disinfection treatment or an intervention, those cells can and over time will start to grow again, meaning that they can spread in the food production environment again, or in the worst case, even multiply within a given foodstuff, for example, and then represent a real danger. So especially the tolerance and the fact that the cells can leave the state again together makes them a big threat or problem in the food industry. And that's why also in the past, not only my research group, but also other research groups have started to really look with new methods into the presence of VBNC bacteria in various food production facilities and different foodstuffs. And some people have started to claim that this VBNC state can really threaten food safety because it's such a fundamental problem for our current monitoring systems, especially. So for me, much more important, although all of this is definitely to a certain extent true, we have to also see them much more as a factor in the persistence of bacteria in food production environment, which is often a very big problem. So it's not really a big problem if you never had Listeria and never had any problems, for example, in your food production environment. But it's much more the case when we, for example, have food production environments where the same clone survives for a long time. And I know people and the quality managers and the staff has been disinfecting and working for a long time trying to get rid of this contamination, and it never really works. So here we know, of course, that we have biofilm formation, we can have disinfectant application failure, maybe biocide resistance. However, I believe that the VBNC induction and also the resuscitation from that state is one of the big ways how bacteria can persist in food production environments and can explain much or many of the findings of finding the same strains, the same isolates for a long time at the same places without being able to really effectively eliminate them. So now I'm going to be quite quick here. This is much more a short overview of methods. I will not go completely into detail here. I just wanted to include them also to be downloaded afterwards, because I just want to summarize that we are actually nowadays having a lot of different techniques to measure those parameters I was talking about. So we know that cell proliferation is an easy and a very easy to understand and to assess viability parameter. However, we can measure all those other parameters, especially integrity and metabolism, nowadays very precisely with a wide variety of methods, and I just wanted to include some of them here. Also, will not go into much detail. So just to mention, I summarized everything here. So we can measure cell proliferation, of course, on the one hand with culture systems, but also alternatively with microscopic systems nowadays. For membrane integrity, we have a lot of different methods available using either microscopy or flow cytometry, or also viability PCR to measure membrane integrity, and also for the active metabolism. We can measure de novo ATP production, we can measure protein production, and many others, which in combination gives us a really viable and a reliable picture of what is really going on with bacterial cells, especially under stress. So how have researchers used this in the past, or which methods have been already established? For example, to measure also disinfection efficacy or resistance. So for example, in the case of Vibrio, we have this amazing assay for quantitative resuscitation, meaning we can compare the colony forming units before and after leaving the VBNC state. So this has been used in the past really to demonstrate, for example, the failure of antibiotics to kill cells in the VBNC state. However, unfortunately, this is only available right now for Vibrio vulnificus and has not been established for other bacteria. This you might be more familiar with. It's the classic fluorescent stains. Very popular is the Backlight live death staining, where we use two different stains to measure the intactness and the integrity of the cell membrane. And this can be either used directly with microscopy, also or in combination with flow cytometry to be used to quantify those cells. And it's a very easy method. It's very established also for the detection of VBNC cells, particularly for detecting Legionella in water. However, unfortunately, in most food production environments, it is not really possible to use this method due to the food matrix, or also if we have a too high flora of actively growing cells of other species, of course, then unfortunately this method cannot really be used. I want to mention also the possibility to use viability PCR, which is also a quantitative PCR system measuring actually the cell membrane integrity. Not going into detail here, but this has also been very nicely established for different pathogens. And the advantages here is that we nowadays have good experience with qPCR and PCR assays also in the food industry. However, the development of viability PCR methods is still quite laborious and also hasn't been really established under real life conditions. And the last is to measure cellular energy or an active metabolism. And these methods are, there's a variation of methods here, and also my own group, we have been working with those methods quite nicely because they're also they give you a great readout and you can make a lot of experiments. However, I have to mention that the con here is definitely that we cannot use them in food production environments because, of course, we have so many other bacterial cells in our systems that we cannot measure something or use something as ATP production, at least as a reliable indicator. However, other groups have started to use the expression or the activity of very specific enzyme systems of bacterial cells, especially pathogens, as an indicator. And this seems to be a very reliable method where we can use the activity of a specific enzyme of, for example, Listeria to demonstrate that those cells are not only present, but also active and viable. Yes. So coming to my first little summary at this point. So what I want you to realize at this point is that we have to realize that Listeria and also other pathogenic bacteria can enter dormancy states. And this can happen spontaneously, but much more important is that it's a response to stress. And in those dormancy states, cells are not culturable, not detectable under routine conditions, meaning that the media we're currently using and the enrichment media especially will not be able to detect the cells or to allow multiplication of those cells when in this state. And the cells in the state they don't have to be, but they can be highly resistant against antimicrobials or physical stress, and that this resistant phenotype in the end allows bacteria to survive and to persist in food production environments, because we have a combination of a high tolerance and especially much higher tolerance against a biocide, for example, than we would expect, combined with the fact that we cannot measure our efficacy of our interventions because our methods are not working. This combination is really what I believe leads to a lot of persistence of Listeria in food production environments. However, they can be detected. And nowadays, the science community has provided us with a lot of different opportunities and possibilities how those cells actually can be detected. So this brings me to the however. So if all of this, what I have been saying and telling you, is not only true in the laboratory, but also of practical relevance, the question, of course, arises, why hasn't this been a big problem before? Or what is really stopping us from going into more detail here? And here I want to mention that we still, also in the scientific community, we still do not really understand how bacterial cells regulate the cell. How do they regulate the entry or the resuscitation from dormance? What really makes those cells so tolerant or so resistant against certain biocides, and what is really the risk according to virulence? Because actually, and this is also unfortunate, it is very, very difficult to study cells in this dormancy state because all our classic also analysis methods in the laboratories normally rely on having culturable states, state cells, or having at least high numbers of those cells. We also don't really know how comparable are the results between species, because this community and this research topic, although there has been a growing body of evidence for a plethora of species, so far people have not been really trying to compare the results between species. So sometimes it's questionable if the result we obtained, for example, for E. coli or Listeria, really transferable. Which also leads me to the question if there's only one VBNC state or if it's even more complicated. So those are the scientific problems we have. However, also on the practical side for the food production companies and food producers, we have the problems that, we are at the moment lacking the analytical tools to really detect and quantify VBNC cells. So while in the laboratories, we can, of course, do a certain amount of experiments and showing, for example, an induction by for example, a heat treatment step or a certain pH shift, we are not really able at the moment to follow or to detect and investigate this under real life conditions, because we're simply lacking tools which are working under the conditions of food production environments, meaning we're having a high background flora of other bacteria. And also, therefore, we do not really know if there are certain process steps or conditions in the food production environments which actively induce bacteria to enter the VBNC state. And as we don't know that, we also, at the moment, we have no real idea how to reduce the persistent cells in the VBNC state. So far here, the research has been much more problem-oriented. We've been reporting increase of tolerance, for example, against certain biocides. But so far, what is really lacking is a good research effort to identify the biocides, the disinfectants, which are still actively or efficiently working against cells in this state, which is also brings me to the topic of the control, because this is at the moment just an unfortunate fact that we have no effective measures. We know that cells can enter the state, but we still have a lot of research, scientific questions, but we have also the problem that we have no research efforts so far to really find solutions for this problem instead of only describing it. But here is, at least from my perspective, the bright side in this field. So however we look at dormancy, we have to realize that this is not an emerging or new threat. It is not something like we have in the antibiotic resistance field, where we believe or where we know that the misuse of antibiotics leads to increasing numbers of resistant bacteria. No, bacterial dormancy has been around forever, at least in our human time scale, of course. So they have been around before, meaning that it is part of the invisible bacterial burden we already had. It is something we can still explore. It's a field where we can still improve on our existing performance. If we have no problem so far with Listeria, for example, in a food production environment, the VBNC state is not a threat. We will not probably find a big problem with VBNC bacteria here. However, it can be an explanation or a realization which allows us to tackle existing problems, which have been there already. So if I want to summarize it again, I would say that Listeria can enter dormancy state in which they are highly resistant, which again allows them to persist in food production environments, and that combining the appropriate and validated methods, we can, as I said, we can detect nowadays which disinfectants are effective or which interventions are effective, and we can also detect those cells potentially. But in the end, what we are still needing is meaningful detection methods we can use in real life environments. And as my personal take-home message in the whole topic is that, as this has been around forever, the realization of bacterial dormancy shouldn't be seen as a threat, but just as a potential start to increase and to really reduce already existing problems. So this is not something which is coming up and represents a new threat, but it's much more a tool or like a different viewpoint on how we have to look on bacteria surviving in food production environments and how we can further improve our current measures and our current systems dealing with those problems. And I believe that if we realize the complexity of bacterial lifestyles and acknowledge that not always everything, that's true for detection methods, but also for interventions or disinfectants, not everything is working all the time. But in the end, realizing this will only make food production more safe and will make food more safe, because it will just eliminate the last pockets of those bacteria in the food production environments. Yeah, and with this take-home message, I just want to quickly acknowledge all my partners, my institute, of course, all the people who have been working with me on this topic in the last couple of years, and also all the people here at our institute, which have been very instrumental in realizing all the research we have been doing in this regard. And I'm really looking forward to answer every question, so I will try to answer every question that you can throw at me, because I know this is a very unusual topic still, and I hope I could shed a little light in the topic of dormant bacteria.
Thank you very much, Patrick, for such cool presentation. And I see that our chat is already exploding from the number of questions. So just to let you know, guys, that we have just 20 minutes left until the end of webinar. Anyway, if we will not cover all questions during the live session, you will find out the answers either on our LinkedIn page or at YouTube under the video recording. So let's start with the first question from Julian Ixen. How do VBNC cells react to the fluorescent live/dead staining with probodium iodide /CeTO9?
Yes, so this is a classic live-dead staining. In the end, both of those stains interact with nucleic acid. The trick of the backlight kit or all those double staining method is that, if I'm not, I might be mistaken here, but I believe that normally CeTO9 can, or what one of the two dyes, I'm sorry to be not on my exactly correct here, but I think propedium iodide normally cannot enter cells if they have an intact membrane. So if the membrane has a damage and a leakage, then those molecules can enter, and then we get the red staining of the cells. So this is the trick. So this is why it's not a live dead staining. This is just very good marketing of this company for many, many years. It's just telling me if the cell membrane is damaged or not. And as both of them interact with nucleic acids, and VBNC cells have to have all of their nucleic acids inside their cell, you will see that the VBNC cells will just look as normally growing cells in those kits or with those methods.
Okay, thank you, Patrick, for answer. And also the next question from Julian is, did you also investigate the mono enters and VBNC state when exposed to dry non-surfaces such as stainless steel?
Personally, we did not investigate this already. I know other people have been doing similar studies with especially Salmonella and E. coli in the past and could find some evidence for Listeria. And we only have been investigating the induction of VBNC cells in response to disinfectants mostly, or for some controls like starvation conditions. But it would be definitely feasible to investigate this, yes. So I cannot give you a good answer. We haven't studied it myself, and I'm not aware of any study with Listeria, but it should be easily doable.
Okay, thank you. Next question comes from Sinfam. I hope I pronounced it properly. And the question is, can we do enrichment culture by BHI medium before do traditional spreading on normal culture media?
That's a good question, and I will rephrase it a little bit, because my answer would be no. Initially, but the topic is very important, because what I mentioned before, the VBNC state is defined by a lack of culturability on a routine media. And this is normally because we do not understand really on a cellular level what those cells need in that specific moment. So this has been done in the past for other bacteria, and there have been a lot of approaches for investigating like persister or other dormancy or dormant cells, and they could show that cells are actually much more sensitive, for example, to hydrogen peroxide while in this dormancy state. And then people were able to manipulate or change the composition of the enrichment media to make them culturable again. So in the end, it could be well possible, and it's one of our research goals as well, to understand the VBNC state well enough to then modify an enrichment media which always is able to detect them. So then we would eliminate the VBNC state per se, but it's just because this definition is a little bit artificial, made by us, and uses the distinction of routine media. So I know BHI medium, we worked with them before with other, we tested several media, will not work, but it is definitely a goal to modify and to improve enrichment media to also be able to detect those cells again. Yes.
And then it will be a topic for our next webinar with Patrick.
I hope so.
Yeah, next question from Julian. Has the behavior of VBNS and mono in the ISO method 1/Fraser ALOA been investigated?
No, unfortunately not. So because it's such a problematic field sometimes when we are talking about not dormancy, because for all legal purposes, if something is not growing on our routine media, we will not be able to cultivate or to detect that. So we would consider them, or what we called in the past when we used molecular methods, or we detecting dead cells. So based at the moment for the ISO method, there is no standard investigating persistent bacteria. This is starting to change in the pharmaceutical sector, actually. And if you look, the Japanese government has started a process to investigate and to change standards there, not for listeria, of course. When we talk about pharmaceuticals, it's a little bit more broader, but to have the more dormant cells also covered by their methods. But for the food or for the classic ISO methods, no. So at the moment, we would know if we have VBNC cells in a normal half-Fraser enrichment, we would simply not see them.
Thank you, Patrick. Next question is from Lucas Richter. Do typical cleaning agents used in food factories based on quaternary, amines, chlorine, etc., destroy VBNC cells?
Yeah, so this is one topic we are doing a lot of research on, and we are very interested in, because as I showed some of the data from our studies before, we could, for example, show that quaternary amines like benzalkonium chloride can destroy VBNC cells for listeria, but you need 100-fold higher concentrations. However, this is a little teaser, I cannot tell you exactly why. If you look and if you screen more molecules, this increased tolerance and resistance is not true for all substances. So also in our publication, we could show that, for example, heat treatment worked against the VBNC cells with the same efficiency as for normal culturable cells. So the cells were not able to survive higher temperatures for longer time in case of listeria, but for quaternary ammonium compounds, for example, yes, they did. But the goal, of course, is for sure to really find those compounds who are really working against cells in a dormancy state. And then if we have evidence for that in our food production facility, to include this in our cleaning and disinfection cycle.
Thank you. Next question is coming again from Julian Isen. Question is, if VBNC cells are inoculated in food, do they switch at some point to the culturable state again?
So for food, for this, I have to be a little bit more specific. For listeria so far, the only really reliable system where you can show good, efficient resuscitation was actually an embryonated chicken eggs. So that works quite well. Then the cells actually completely return to their culturable state. For food, this has not been shown. So we don't really know if in the food matrix or in which food they would resuscitate. But again, as we are not really understanding what's going on with the phenotype and what those cells really need, what we monitored is that they stay within the food for a long time and they stay in the VBNC state, but we couldn't observe any reactivation again at that point. but this might be different and for other bacterial species. And as I said, especially the studies showing that it works quite well with embryonated chick eggs could also indicate that it would still be a problem for the host in the end.
Thank you, Patrick. And next question comes from Thin Farm. Also, does VBNC impact to European regulation 2073/2005 on enumeration?
So again, the easy answer here is no, because at the moment they're not considered viable regarding any legislation. So if we would use a method which can detect them, which at the moment, for example, would be a classic PCR system, we would consider that a false positive. And this is exactly where the whole topic of VBNC has this potential to change and I believe, to make changes, because we have to realize that not all of those were false positives, but they were actually correct positives, but not viable, not detectable. So they're not really impacting the regulation and enumeration right now, because there's no way to enumerate them efficiently. So at the moment, no, but we're working on it.
Great. And I would assume that it was last question for today. And again, guys, this video anyway is recorded and it will be published to our YouTube channel as well as to our LinkedIn channel, Nemis Academy. And don't hesitate to follow our LinkedIn page and register to our upcoming webinar. And the next one will be already this Thursday, led by Jeff Banks, and it will be about root cause analysis. And the link to this webinar you will be able to find on our website as well as on our LinkedIn page. And I'm very happy to thank Patrick for time given today and for this valuable information. And wish you all great day, and stay tuned with our future webinars. Ciao all.
Thank you very much.